judges, since I am an attorney myself, I have never introduced a world-renowned scientist and president of MD Anderson Cancer Center. So in preparation, I did what any self-respecting attorney and compliance officer would do. I watched the Colbert Report. I'm joking about that, but if you have not seen Dr. DePinho's presentation on the Colbert Report, I recommend it, especially if you are not a scientist. It's a great explanation of his work on the study of telomeres and how they affect the aging process. As I am rapidly approaching my 45th birthday, I am most interested in his science and how it will affect my beauty regimen. More importantly, however, I think we are all interested in how his science affects the disease process and its effect on cancer in particular. It was almost exactly 1 year ago that Dr. DePinho joined us as our 4th full-time president. He came from Harvard Medical School, where he was a professor of medicine in the genetics department. He was also the director of the Belfer Institute for Applied Cancer Science at Dana-Farber, where he worked to reverse the aging of mice. He attended college at Fordham in New York and medical school at the Albert Einstein College of Medicine. He has published numerous publications and received countless awards. So many, in fact, that his stunning CV is 35 pages long. We are indeed honored to have him here with us at MD Anderson and here today. Now, it is my honor to introduce Dr. Ronald DePinho, and don't be fooled by his appearance, he's actually 450 years old.
Thank you very much. Over the weekend, I have no life, I was preparing for this, and I had all these scientific slides, and then I realized that the audience was a little bit different from the kind of folks that I talk to when I talk about aging science and so on. And I see that there are people from my lab here, so get back to work. But it is an incredibly exciting time in the history of who we are as a species, because for the first time in our existence, we actually have the ability to understand who we are and do something about it. Now, we've done this to a certain extent over the last 70 years. For the last 70 years, thanks to hygiene, the germ theory, improvements in surgery, vaccination, antibiotics, and so on, we have gone from a worldwide life expectancy, driven in part by some of the great wars, of 42 to today about 74 worldwide and rising. So that's really something to celebrate. It's a great accomplishment that we essentially were able to double the life expectancy of who we are, which is extraordinary. And much of that, again, relates to, washing your hands and some vaccinations and a variety of other things we've done. It's not a lot of necessarily the fancy, schmancy stuff we do here at this institution, which clearly has an impact on specific patients. But when we think about the future is going to be one that's going to be, I think, one of tremendous opportunity and extraordinary challenge. You think about having life expectancies continue to rise. And so by 2025, there are going to be 1.2 billion people over the age of 60 worldwide. So what's the significance of that? From a healthcare standpoint, the significance is that after the age of 60, every 5 years, the incidence of the great 4 diseases, which extract around $1 trillion in today's economy, that is Alzheimer's, diabetes, heart disease, and cancer, about $250 billion each. Those diseases increase, they double every 5 years. By the time you're 85, you have a 45% chance of having Alzheimer's. If you're a man, you have a 1 in 2 chance of having cancer. A woman, 1 in 3 chance. So those are the facts. So there's this very, very intimate link between advancing age and those diseases. And the question that I've been interested in trying to understand is, what is the basis for that intimate link? Is it just true, unrelated? or if you understand the molecular circuitry of heart failure or diabetes, how much is it linked to things that actually accelerate and cause aging? Are the two really two sides of the same coin and intimately linked on the molecular level? And I think the answer to that is now exceedingly clear. It's yes. And so this provides tremendous opportunities because essentially what our children are going to inherit is a world that doesn't have a sustainable business model. You know, when you think about all of these people coming online at north of 60 and having to have diseases that extract a huge impact on society. You look at Alzheimer's. Alzheimer's today, we spend around $200 billion in Alzheimer's today. We have 5.4 million people with Alzheimer's in the United States alone. By 2050, if we don't do anything about the disease, there's going to be, we'll spend, in today's dollars, $1 trillion just on that one disease. So we have to ask ourselves, can we prevent those diseases from happening? And maybe even more so, actually fundamentally impact on the aging process itself. Now, having studied this disease of aging, and I consider it a disease, I don't consider it to be an inevitable aspect of just advancing in years, I think that we're in a position to not only impact on aging itself, but impact on age-related disease itself, and try to understand the biology of aging to a level that we can identify those individuals that are at risk for disease, that might undergo particular kinds of preventive strategies, screening strategies, and so on, to really diminish the impact, economic, political, societal, of these age-related diseases that are just going to extract a huge toll on future generations. So I'll tell you the story of how we got into this. So it was 1994, and this woman scientist calls me up, Carol Greider, who was a young faculty member at Cold Spring Harbor, and she told me about this stuff called telomeres. You know, I known about telomeres. Telomeres are the ends of chromosomes. So your chromosomes have to maintain their integrity. Right? So you have caps. They're like the parts of your shoelaces where you have caps. You want them to become frayed. What happens with aging is that telomeres become damaged with time. They become shorter as cells divide, and they also accumulate damage. And they accumulate damage that doesn't get repaired because the ends of the chromosomes are a privileged part of DNA where you actually don't want to have repair machinery act on those ends. Because if they did, your ends would fuse, all your chromosome ends would fuse. You wouldn't maintain those packets of chromosomes. So that's how it solved the problem. Evolutionarily, no problem. And it's not a problem of evolution because, you know, when we were running away from saber-toothed tigers and woolly mammoths, it didn't really matter that no one, you know, would live to be 60 or 70. So you'd have this evolutionary pressure for things that are important early in life, but not necessarily late in life. So these telomeres are designed to maintain chromosomal integrity, and they accumulate damage. And there were lots of theories back then that maybe that damage, when it becomes damaged enough, you get chromosomal instability that might cause cancer, or maybe the damage accumulates to a point where it precipitates the aging process, and so on. So Carol Greider called me up and she goes, you know, I just identified the gene that encodes for an activity called telomerase that allows the ends to be formed. And telomerase is normally off in our cells as adults. It's on and active and in our germ cells. That's how we propagate the species over millennia, to maintain the telomeres from generation to generation. But in adult tissues, that enzyme is off. It's at very low levels. So, but in the mouse, curiously, it has some expression, etc. And so she said, you know, if you can help me knock this gene out in the mouse, we can then study these engineered mice and ask, what happens? Do you get accelerated cancer, or maybe less cancer? Do you have precipitate aging and age-related diseases and so on? So it was an ideal system in which to ask this very fundamental question about the role of telomeres in cancer and aging. So that was in the 1990s. We knocked out the gene. People in my lab studied it. They're now all famous scientists, left me behind in the dust. And Carol Greidel went on to win the Nobel Prize a couple of years ago, along with Liz Blackburn and Jack Shostak for their fundamental work in the discovery of telomeres and telomerase. And so that was all very exciting. And so what we did is we actually tried to understand the role of telomeres and telomerase in cancer and aging. When we knocked out telomerase so that the telomeres became shorter and eroded and became very damaged, what we found was these animals aged prematurely, so it proved that you have to maintain telomeres in order to prevent aging, that could be an important instigator of the aging process, and the animals got more cancer. And that gave us the insight that what happens, why you have this intimate link between age and cancer, is that you have these eroded telomeres over time that then create instability of your DNA, reshuffles your genetic deck, and you might pull out the cancer card for that cell. And so all of these things gave us fundamental insights into the role of telomeres. So now that we had this model organism that we can manipulate and study on the molecular level, we started asking more fundamental questions. So we actually engineered a mouse where we could turn the gene on or off, so that we gave that mouse an artificial chemical. It would turn telomerase off. In the off state, they age prematurely. And if we turned it back on, we would prevent aging. And so that was a very useful model. We also asked, if we cause damage in telomeres, what is the molecular circuitry that precipitates the aging process? Right? We had a perfectly controlled model system in which we can damage telomeres, get aging, and ask what happens in between. What is the connection that ultimately causes senescence in cells, or heart failure, or brain health failure, etc., or thin bones? And so that analysis, which was done by another gifted trainee in my lab, who's now a faculty member at Baylor, he basically looked at the heart, the brain, the blood cells, and so on, and he asked, is there a molecular circuitry that gets tripped? Can I identify the molecules that are actually responsible for carrying out the aging process? And what he found was that indeed there was, that there was this common molecular circuitry. And it was a circuitry that involved the damage of DNA, that is, the eroded telomeres, the activation of a molecule known as P53, which senses damage, then the repression of another molecule that, there's going to be a test on this afterwards, another molecule that turned out to be the master regulator of mitochondria. Who knows what mitochondria are? Mitochondria are the batteries in your cell that make ATP. They are essentially the critical engine that generates the fuel for your cells to operate. And so it's long been recognized that mitochondria become fewer and not as effective in producing energy as you age. And so that's one of the reasons why, if you're on a treadmill and you're my age, it's 220 minus your age for maximal cardiac output, because your mitochondria can't make enough ATP to make your heart beat as fast as it was when I was 12. And so that's a reflection of mitochondrial decline. And so the question is, no one had ever connected the accumulating damage of DNA that occurs in aging and the functioning of mitochondria. So that linkage of those molecules, of telomeres, P53, to this master regulator of mitochondria, to mitochondrial decline, which is also important for protecting us against free radicals, serves as a sink for free radicals, led us to identify this core pathway for aging. So we now knew the molecular circuitry. And the reason why that's important is that circuit is very important for precipitating the aging process, but it also turns out to be that exact same pathway is important for diabetes, it's important for heart disease, it's important for cancer, and it's important for neurodegeneration and Alzheimer's. And so that's why you have this link between advancing age and cancer in those diseases, because the same molecular circuitry that causes aging gets affected, goes awry, and causes these age-related diseases. So that's why there's this intimate link. Okay, so that's the background. So you don't have to remember all of this, but just remember that there were a lot of different theories of aging a few years ago. Accumulating DNA damage, free radical theory, and mitochondrial decline. So what this study told us is that there's a single molecular circuitry that governs those three processes. Having identified that, it then gave us the notion that maybe what we can do is try to manipulate that circuit so that we can prevent aging or reverse it if it happens. Now, much of the work in aging, which has studied this pathway or components of that pathway, has really focused on trying to slow the aging process. And that's been pretty effective in model organisms, and it's been proven to be quite impressive in its increasing life expectancy and even lifespan in lower systems. But no one had ever asked the question, could you reverse it? No one, including us, felt that this was possible. So we went back to this mouse where we can toggle the telomerase on and off. And when in the off state, the animals prematurely aged. And I mean, we got them all the way to a point where they were really at death's door. Another month, they would have died from old age. And so this would be the equivalent of somebody in their 90s. So they had shrunken brains and impaired cognition. They were infertile. If you look at the testes, they had barren seminiferous tubules. You could barely see the sperm. They had gray hair and a kind of looked shabby. They really looked old. So in that state of severe degeneration, we flipped the telomerase switch back on. And what we were expecting at the time was a slowing or stabilization of the process. And what we found was a Ponce de Leon result. That these animals that were like 90-ish suddenly became like Allison. And they were youthful and vibrant, and their brains increased in size, their fertility was restored, the coat hair was restored to a healthy sheen and shine. So that taught us that tissues, even in a very severe state of degeneration, retain a remarkable capacity to renew themselves. So when I look at the aging process now, and I think about what is it that we can do, I think that if we could really understand this core circuitry and manipulate it, there's no reason why aging could be at least slowed, if not reversed. And as a result of impacting on this core axis, we may actually also prevent these grade 4 diseases, which would be the holy grail, right? That you actually hit it off at the pass, that the fundamental circuitry that's involved in aging, and as a result age-related diseases, is maintained in an intact state in such a way that you actually don't get some of these side effects and so on, or if they do occur, to actually reverse them. So one of the things that we now know as a result of this core axis is we ask the question of things that we know to be important for preventing aging or slowing it down, like exercise, caloric restriction, stress, and so on. Are these things tying into this core pathway? And the exciting news is yes. That is, these molecular circuits, the signaling through that pathway is affected by exercise. So a very famous study done here at MD Anderson showed that, and I'm not getting into the advice part of my talk, that the 15 minutes of vigorous exercise several times per week can dramatically increase life expectancy by as much as 3 to 5 years as a single intervention. Right? We know that if you are not obese and you control your diet, and that also has a huge impact on the signaling flux through this pathway and the incidence of heart disease, Alzheimer's, diabetes, and so on. Cancer for sure, right? So those are things that you actually can control. Of course, smoking causes damage, and it causes damage at telomeres, and that also precipitates all of these age-related diseases. So, there are three things right there that if you actually, where you have control, as a population, we were to really pay attention to, and that is, have exercise, proper diet so that we have, proper BMI, and smoking, that could have a huge impact on this signaling circuitry. The other one is stress. So stress, you have to define stress. Acute stress, which is exercise, so this morning I exercised for 25 minutes, that was acute stress. That's actually good for you. But chronic stress isn't, like being president of Indiana. Chronic stress is seriously, it would be like a mother who has to care for a young adult child with schizophrenia and worrying about just, having just around the clock chronic stress of concern about the health and well-being of that child, right? That is chronic stress. Or the loss of a loved one, and so on and so forth. That, what happens there is that you have hormones in your body that increase, these stress hormones. And we now know these stress hormones engage circuits that decrease your ability to defend against free radicals. So you have more free radicals in your cells. More free radicals in your cells chew telomeres. Telomeres cause this whole process to get tripped and accelerates the aging process. You get cancer, diabetes, and a variety of other things. So, when you think about healthy living, and I'm passionate about this with our employees, really, from looking, I did a walk through the cafeteria the other day and looked at the type of food we were serving. I talked to Bill Bond on a regular basis in terms of trying to get us to exercise more, etc., nutritional support, etc., stress management. These are all things that are really fundamental to healthy living. And they have to be part of your DNA. They really have to be part of the way that you approach. And just like we would never think of not going a day without eating or a day without sleeping, not exercising or managing your stress in some positive way is also not an option, because those things profoundly impact on who you are, your ability to operate as, here as an employee or a parent or, a son or a daughter and so on, for those around you. So I want to do everything that I possibly can to help with your well-being. I think that science in the coming decades will deliver on some very exciting advances that may help us prevent or reverse things like obesity or improve brain health by understanding neurodegeneration and a variety of other things that prevent, obviously, cancer, all of these things. But today, before those game-changing conceptual breakthroughs and future Nobel Prizes are delivered to us as a species, listen to what your mother told you growing up, which is exercise on a regular basis, have a healthy perspective on life, and try to live a full and rewarding life that allows you to really feel good about who you are and help those around you. Thank you very much. So I'm not quite sure if that's what you were expecting, but here you go. I'd be happy to take any questions or talk about anything else. Pardon me?
Forgot your bags.
That's part of the diet. Actually, for food, so that really, key foods. Obviously, it's the amount that's critically important. But things like broccoli, very important. There are certain fruits and vegetables that are extraordinarily high in antioxidants. So pomegranate is extremely powerful, and in fact, there's a very good study that shows a dramatic reduction in prostate cancer in men. And I mention this because you would think that vitamin E, which is an antioxidant, would do the same thing. And yet pomegranate has this dramatic reduction, and we just recently learned that excessive vitamin E intake actually increases prostate cancer. So taking things out of their natural context may somehow destroy the properties that really enable it. So blueberries, pomegranate, broccoli, other foods that are high in antioxidants, vitamin C, and so on, are really good for you.
The mouse that you reversed his aging, you said he was in his 90s, so how young did he become 80?
No, well, basically, it's a little bit rigged. So the animals were sort of the equivalent of somebody in their 40s or 50s, okay, when, in terms of their chronological age. The biological age was the equivalent of somebody in their 90s. And when we flipped the switch back on, they reverted back to that middle-aged youthfulness. They don't become an embryo, you know. It's not like the constellation thing where you become a baby, you know. Basically, this circuitry doesn't influence the development of your cells. It influences the physiology of your cells.
Like, for example, your muscle mass.
Yeah.
In other words, does it get better, it goes up?
Yeah, everything. I mean, you basically, your brain size, what happens in your brain when you age is a couple of things, is that stem cells don't renew and make newborn neurons. It also, the sheath, the myelin around your neurons that make your neurons work well, also become reduced in size. In fact, that's the major reason for the contraction. It's not the loss of neurons as much as it is about decreased myelination. And just 1 month of flipping the switch back on, the myelin was restored, and the neural stem cells started proliferating, creating newborn neurons. So it's very dynamic. You would think that the brain is fixed. And what this study taught us was that it retains this remarkable plasticity.
So you mentioned you looked at the what's being served here in cafeterias and places like that. So I mean you will find plenty of things like the whole soda area, bunch of other things. I mean, is it possible to make these kind of changes that all we offer is water, not any kind of soda? Introduce changes in what's served, food-wise, like something that involves more antioxidant, stuff like more spices. I mean, is it possible to bring about those changes and send a lesson to the whole country that this is what the cancer center is doing?
You bet. So this is happening already, but it needs to happen in a more accelerated way. You'll be seeing some announcements that came as a result of this walkabout that I did through our food services. But what has happened already is that there is a shift in the ratio of healthy food and comfort food. The hamburgers and French fries versus, let's say, salmon and vegetables. And so, and what they've done is to actually experiment to be able to see, to actually shift that ratio and get people to be more, and also even things at eye level. If you notice in the cafeteria, like the bad stuff will be, out of eyesight, whereas the things that will be healthier for you are more in line. And so on. But there are a few things that came about as a result of my walkthrough that we're going to be doing. And I'll be soon sending out messages to raise the consciousness of people, because there's a lot that you can do for you, and sometimes you just don't know, or you just have to be reminded that it does make a huge difference. And I agree with you 100%. If MD Anderson has not set an example, you think about what we did with smoking years ago and set the example as an institution in terms of smoke-free, et cetera. Those are the kinds of things that we basically have to say. We stand for human health, and this is what we're all about.
What is the next stage in research? Will you be continuing to study the mouse model, or you
Well, I think there are a couple of things that excite me. One is, can we pharmacologically manipulate this pathway? Now that we actually can follow it and measure it and see if we can reverse, like enhance mitochondrial biogenesis and function. Are there drugs that can do that? So that's one aspect. And the other one is to actually assign risk. You know, we say, is somebody, just because they're 60 or they're 80, where are they on the biological plank? You know, in terms of, you know, being very close to getting these diseases. So maybe if we can monitor, and I'm working with Shifan Wu and others here in the Epi department to try to develop better biological risk models. That, you know, it's not, you can't just look at telomeres, you can't just look at mitochondria or certain genes, but if you look at all these things together, they could provide extremely robust risk models that if, even though you might be 65, if you've got the biological age of somebody in their 80s, you may need to be surveilled more actively to be able to make sure that you get disease caught early.
Seems like the research is looking,
Can you speak up?
it seems, if I understand it correctly, it looks like the research is looking at maybe a specimen that was healthy or young and then age. Will there be additional research, or is the research already at a point where it can be applied to someone who's already born into the world with these damaged genes, like a child born into the world with cancer, whereas you're not looking at the child healthy and then aging into cancer,
Yeah.
the child is actually
Already having a problem. Those are, most of the cancers in childhood are really about inherited developmental abnormalities, and so the die is cast in utero for a lot of these children. There are certain age, you know, premature aging syndromes, very famous ones where, you know, you might be 20 years old and look like somebody who's 80. Werner syndrome, Bloom syndrome, varieties of very rare syndromes. Knowledge of this pathway may actually provide preventive strategies for those individuals to live longer and have anti-aging. And if we develop something like, you know, a drug, that would be the first people in which this would be tested in, I would think. So I think that for a lot of the it's a little bit different with the childhood diseases because they're more developmentally regulated.
You mentioned about talking back and forth with that mouse, and you mentioned that the myelin had repaired itself, filled itself up. As you know, there are diseases that affect the myelin in human beings, and can any of that at any time be applied to a human being, like a human being with multiple sclerosis,
Right.
who's refractory to medication? Who is now progressive, and could anything like that, I mean, can it be at some point that it's going to progress to where it can be applied to a human being?
So the question is, can this be applied to more specific diseases like MS, because we have these dramatic effects on myelination
Yeah.
And so on, which is a disease of the myelin. And I don't know the answer to that. It's possible that this study of how myelin gets, regenerated and so on could help. But for MS, its molecular pathogenesis may be different. You know, there's this theory about the immune system playing a role, etc. So you may be able to enhance myelination through study of the mouse that we created. But I suspect that it wouldn't hit the nail on the head necessarily with respect to that disease.
Because the T cells would keep on attacking.
If that's the basis for the disease. You know, with MS, one of the great challenges that we have is we're still, going across the Atlantic Ocean, as Christopher Columbus did, and wondering whether or not we're going to fall off the edge of the earth. We don't really know where we are with that disease, from my perspective. Not being an expert, but I think that there is a lot more that needs to be understood about the disease for us to be able to go after it. It's almost like cancer 25 years ago. We didn't really, had some ideas, but we really were flying blind as opposed to today.
Thank you.
I think the article that was sent out for the notification for this week mentioned something about a potential increase in cancer rates coinciding with an increase in telomerase activity. Has that, and I think somebody mentioned a point about how the reduction in senescence may counteract that.
Yes.
Has that kind of been quantified, or is this sort of?
Well, I think if you're going to reactivate telomerase, which is important for cancers to be fully progressed, well, this is where you have to be very careful about doing no harm. That if you have an initiated cancer in that person, let's say a person's 50 years old and has early stage prostate cancer or breast cancer, and you flip that switch on, you may actually fuel the progression of that cancer. So you've got to know, I think if you do this, you have to do it in a far more preventive strategy, because if you sort of do it very late in life, you may actually precipitate the development of cancer. Having said that, in the mice that we've reversed the aging process, we did not see any increase in cancer. So that's kind of good news, but it is something we have to worry about.
Well, actually, on that note, so it sounds like these specific tests were done in mice that were artificially aged, I guess, by knocking out telomerase
Yeah.
function. Have these tests ever been performed in mice that have naturally